An antibody, also called immunoglobulin, or Ig, is a protein produced by the B lymphocytes, or B cells, of the immune system in response to the presence of a specific antigen. Antibodies bind to antigens and help neutralize or destroy them, providing protection against infections. Antibodies are Y-shaped proteins made up of 4 polypeptide chains, 2 heavy H chains, which are larger in size, and 2 light L chains, which are smaller in size. These chains are held together by disulfide bonds. The variable region, or Fab region, at the tips of the Y shape is antigen binding and is unique for each antibody. The constant region, or FC region, determines the antibody's function and class. Key feature: each antibody is highly specific for a particular antigen, like a lock and key mechanism. The human immune system produces 5 classes of antibodies, or IgG. IgG, which is the most abundant, provides long-term immunity and crosses the placenta to protect the fetus. IgA is found in secretions like tears, saliva, and breast milk, and protects mucosal surfaces. IgM is the first antibody produced during infection and indicates a recent infection. IgE is involved in allergic reactions and defends against parasites. IgD helps in B cell activation and is found on immature B cells. Neutralization. Antibodies block the activity of viruses, bacteria, and toxins. For example, anti-rabies antibodies neutralize the rabies virus. Neutralization. Antibodies block the activity of viruses, bacteria, and toxins. For instance, anti-rabies antibodies neutralize the rabies virus. Opsonization. Antibodies coat pathogens, making them easier for phagocytes, macrophages, and neutrophils to engulf and destroy. Agglutination, or clumping, occurs when antibodies bind to multiple antigens, forming clumps that are easily removed by immune cells. For example, blood grouping is based on antigen-antibody reactions. Complement activation. Antibodies trigger the complement system, leading to the destruction of bacteria, memory formation. After an infection, memory B cells retain information about the antigen, ensuring a faster immune response in future infections. Vaccines stimulate the production of antibodies and memory cells, providing long-term immunity. For example, the polio vaccine induces the formation of antibodies against the polio virus. Antibodies recognize specific epitopes, or binding sites, on antigens. They are part of humoral immunity, which is a B-cell-mediated immune response. Monoclonal antibodies, which are lab-made antibodies, are used in diagnostics, cancer therapy, and COVID-19 treatment. Antibodies are the first line of defense against infections. They are used in blood tests to detect diseases like HIV, malaria, and COVID-19. Additionally, they are essential for autoimmune disease research and allergy treatments.